Our discussion this time is about analysis of variance or ANOVA. Analysis of variance or ANOVA is a statistical analysis method that belongs to the branch of inference statistics. In Indonesian literature, this method is known by various other names, such as analysis of variance, variance analysis, and analysis of variance. ANOVA method was first introduced by a statistician named Ronald Fisher. ANOVA analysis is usually used in experiments with the principle of comparison between two or more groups, which in practice, ANOVA also considers the diversity of data which is manifested in the value of the variance. ANOVA function. The ANOVA test is used to compare the population averages to find out the significant differences between two or more data groups. There are two types of ANOVA test, namely one-way ANOVA test and also two-way ANOVA test. One-way ANOVA is a test technique that only takes into account one factor that causes variation. So, although in practice the method is used to test two or more groups, however, there is only one factor to consider. Two-way ANOVA test is a test method that considers two factors that cause variation. Two-way ANOVA is often used by researchers to overcome the independent variable consisting of more than one group. This is the differences between one-way and two-way ANOVA. One-way ANOVA is used to test the comparative hypothesis of the average k-samples, if each sample consists of only one category. Meanwhile, two-way ANOVA is used to test the comparative hypothesis of the average k-sample when the researcher categorizes the sample. The hypothesis test that allows us to test the equivalence of 3 or more ways simultaneously using variance is called one-way ANOVA. The statistical technique in which the interrelationships between factors, influencing variables can be studied for effective decision-making, is called two-way ANOVA. Types of ANOVA other than one-way and two-way ANOVA. The FIRS is ANOVA repeated measures. That used when in experimental design using research subjects included in different treatments. Related to the example above, for example, the same plant is given fertilizer with different doses. And the second is multivariate ANOVA. In contrast to the ANOVA test which only measures one response, MANOVA measures more than one response in one experiment. For example, we examine the impact of fertilizers at several doses. The responses that were studied were more than one, for example the levels of nitrogen, phosphorus and potassium in their effect on crop yields. ANOVA objective. The aim of ANOVA is to draw conclusions by finding different datasets. This is done by giving the null hypothesis to the data population. Junri Lasman Marpong and friends in the journal Application of the ANOVA Method for Analysis of Mechanical Properties of Coconut Fiber Composites, 2017, state that the null hypothesis is data from a random sample with the same population having the same expected mean and variance. When testing with the ANOVA technique, you must also be able to calculate test statistics. This study aims to test the hypothesis whether the compared groups have similarities or not. The final result of the ANOVA analysis can be seen from the calculated f-value. This calculated f-value will later be compared with the value in the f-table. If the value of f-arithmetic is greater than f-table, it can be concluded that the ANOVA analysis is high or the analysis is accepted. On the other hand, if the value of f-table is smaller than f-arithmetic, then the results of the ANOVA Test 9 analysis are rejected or HO. The principle of ANOVA is to analyze data in two sources of variation, namely variations within the group or within, and also variations outside the group or between. If the variation within the group is greater than the variation outside the group, that means that the compared mean values have different effects. On the other hand, if the value within is smaller than between, so it means that the intervention did not give effect or difference. This is the ANOVA assumption. The first is observation independence. When planning to perform statistical tests with the ANOVA principle, it should be ensured that each observation included in the analysis is independent. The second is normality. The data used in the ANOVA analysis should be normally distributed data, because the data will later be used in the statistical test F. And the last is homogeneity of variance. Homogeneity of variance means that the variance between the groups being compared must be homogeneous. This is often known as homoscedasticity. This means that only one estimator is used as the variation in the sample. Conceptual foundation of ANOVA. Like the t-test, even in the ANOVA test you have to calculate the test statistic, in this case the F ratio, to test whether the groups being compared are similar or not. The statistical language of the ANOVA test hypothesis can be written as follows. Ho M1 equals M2 equals M3 equals 0. Usually in the hope that you will be able to reject Ho to provide evidence of that alternative hypothesis, H1. No Ho, to test Ho, you randomly sample groups of participants, sample, respondents and assign measures, dependent variable. Then see if the measures differ significantly for various conditions. If different, then you will be led to reject Ho. As in other statistical tests, we reject Ho when we find that the test statistic measured by F statistics exceeds the F table with a certain level of confidence. Another way can be done by looking at the P value, probability value, which is lower than 5%. For example, we use a 95% confidence level. The principle of the ANOVA test is that we compare the variances of 3 or more sample groups. More than just comparing the mean, average, value, the ANOVA test also considers the diversity of the data which is manifested in the value of the variance. ANOVA in linear regression. Sometimes readers are quite confused by the ANOVA table on the results of linear regression analysis. Of course, if you understand the real purpose of this one test, then you will not be confused anymore. ANOVA in its calculation compares the mean square value and the result is to assess whether the linear prediction model is not significantly different from the estimated coefficient value and standard error. This is the case examples in one-way ANOVA analysis. A researcher conducted a study on the effect of jatropha on chili production. So the research was conducted using 5 kinds of spacing. A equals 15 by 15 centimeters, B equals 15 by 20 centimeters, C equals 15 by 25 centimeters, and D equals 20 by 20 centimeters, and obtained the following data, in quintals or Ha. By using a significance level of 5%, we test the hypothesis which states that there is no significant difference in chili production using 4 kinds of spacing. It is assumed that chili production data is normally distributed with the same or homogeneous variance. Basic decision-making in one-way ANOVA analysis. The first is, if the significance probability value is greater than 0.05, then there is no significant difference in chili production using 4 kinds of spacing. And the second is, if the significance probability value is 0.05, then there is a significant difference in chili production carried out using 4 kinds of spacing. And now I will give you the steps for one-way ANOVA analysis test with SPSS. The first is, open a new worksheet in the SPSS program, then click Variable View, then in the name column type Production in the first row and Treatment in the second row. In the decimals column, type 1 for the first row and type 0 for the second row because the data is in categories. In the label column, type chili production for the first row and treatment for the second row. And the second is, in the second row, in the values column, click the mouse on the small box to the right of the cell. In the value field, type 1, in the value label field, type distance A, then click the Add button. Enter again for value type 2, in the value label type distance B. Click the Add button, click again for value type 3, in value label type distance C. Click the Add button, click again for value type 4, on the value label type distance D. Click the Add button, because it's finished then click OK. And then step 3. Click Data View, then type the data as above according to the variables. Download input data. And then next. Select Analyze, then Compare Means, and then One-Way ANOVA. And the fifth, click the production variable, enter it into the dependent list, then click the treatment variable, enter it into factor. And next, click the options, under Statistics, select Descriptive and Homogeneity of Variance Test, then click Continue. After that, click Post Hoc, from the available options select LSD, then click Continue. To end the analysis process, click OK, and when finished, a display like this will appear, and if it is, then the analysis has been completed. To shorten the explanation, pay attention to the third output only, namely ANOVA. Based on the output above, the significance probability value is 0.044. Therefore, the significance probability value is 0.044 less than 0.05, so the above hypothesis is rejected, which means there is a significant difference in chili production by using the 4 different spacings. And now I will tell you about basic requirements for two-way ANOVA test. There are at least 4 requirements or procedures that must be met before we can perform a two-way ANOVA test to analyze research data or test hypotheses. The 4 requirements are as follows. The first is, standardized residual values must be normally distributed. This is the first condition that must be met before we perform the two-way ANOVA test. Therefore, we need to do a standardized residual normality test first to detect whether the value is normal or not. If it turns out that after the test is done, the results are not normal, so we have to forego the two-way ANOVA test, and switch to non-parametric statistics. Guide. Practice of standardized residual normality test in two-way ANOVA SPSS population. The second. The populations of the data variance are the same or homogeneous. The way to detect it is by doing a homogeneity test. Note. The homogeneity test can be done at the same time when we do the two-way ANOVA test. The third. The samples used are not related to each other. This means that each population is independent of each other within the group. And the last requirement is related to the type of data used, where for the dependent variable data, dependent variable must be scaled intervals or ratios. Meanwhile, the independent variable data or independent factor is on a categorical scale. Thank you guys for watching this video. I hope with this video you guys can understand what is ANOVA analysis and how to do ANOVA analysis with SPSS. So, thank you again guys and have a blessed day.